Now we are ready to do blood testing on simulated blood. Your at-home kit should have supplies that look similar to this: bottles of blood and bottles of antibody. The kit that I have in class has names on the bottles of blood. The kit that they send to you might not have names, it might just have numbers, but the blood types are all the same. So 1 is Smith, 2 is Jones, 3 is green, and 4 is brown. So you can coordinate what you have in your lab kit with what is already written in your binder. So the idea of testing blood, remember blood type is which antigens you have on your red blood cells. Antibodies will bind to antigens. So we have, for example, here, this bottle is A antibodies. When you mix this with the blood, it will look for A antigens. It only binds to A antigens. It ignores B's, it ignores D's. It's like a lock and a key, right? Only the ones that fit can bind. We take a few drops of this, we mix it with the blood. So I've already put the blood into these trays. You stir it up with a toothpick. If this A antibody finds an A antigen to bind to, it will cause agglutination. The blood cells will clump together. And you'll be able to see that the blood will either have little clumps, they'll be like little specks of pepper kind of floating on the blood, or the blood will get cloudy if agglutination occurs. So if that happens, if you get agglutination, then that means that the A antibody found A antigens. So that person has A antigens. If you mix the A antibody with the blood and nothing happens, then that means that the A antibody did not find any A antigen. So that person does not have A antigens. So what I'm going to do is, you can see these trays are labeled with an A, a B, and an RH. I have all three antibodies, A, B, and D antibodies. I'm going to put a couple drops of the A antibody there in the well labeled A. I'm going to put a couple drops of B antibody in the one labeled B, and a couple drops of D antibody in the one labeled RH. Then I'm going to mix it up with the toothpick, and we're going to look at the blood. Okay, here you can watch what happens as we stir. So here's when I haven't stirred yet. As I'm stirring it, you can kind of see those clumps forming in there. So I've done all of them now. We'll interpret the first one together. So remember, this would be bottle number 1 in your Carolina kit. So if you have that kit, go ahead and do that, follow along, make sure we get the same results. If you look closely at the A and the RH, you can see the clumps there. And if you look at the B, there's nothing there. It's just still clear. That means that the A antibody found A antigen. The D antibody found D antigens. The B antibody did not find any B antigens. So this person has A's and D's, but no B's. That is A positive blood. So the other ones, if you have your Carolina kit, go ahead and do them on your own and get your blood types. If you don't have your Carolina kit yet, I will go ahead and show you the results of the other 3 bottles, but you do your interpretation. So this is bottle number 2, Mr. Jones. There's the A, there's the D, and there's the B. And if you're not sure, if it's hard to tell from the video, the B is cloudy, the A and the D are still clear. Here's number 3, Mr. Green, the A, the D, and the B. All 3 of these are cloudy and clumpy. And Miss Brown, the A, the D, and the B. all three of those are still clear. And if we kind of put these together just so you can compare, the one on the left is Mr. Green, where all three of them came out cloudy and clumpy. And the one on the right is Miss Brown, where all three of them are still clear. So remember, the antigen looks for its antibody. If it finds it, it gets clumpy and cloudy. So clumpy and cloudy means that that antigen is there. Clear means that that antigen is not there.